Okay, hi everyone. So today we are going to look at into introduction to forensic serology and DNA analysis. All right. So basically, for this topic, it will be divided into two lectures. So firstly, you are going to look at what is forensic serology and what are the presumptive tests that we are going to do in terms of how are we going to know whether it is human blood, whether it is animal blood, or whether it is blood actually? All right. So, and then we are going to look at on the second part of the lecture, we are going to look at into the DNA analysis. All right. So basically, for this chapter, at the end of this chapter, you should be able to describe or explain the use of blood, semen, saliva, and other human biological fluids in forensic investigation, right? So you also need to be able to know about the presumptive and confirmatory test for human biological fluids. Okay, so now let's look at what is serology actually. What is the definition of serology? Serology is the study of antigen and antibody reaction. Okay, so it is a study of antigen and antibody reaction. Okay, the study of biological fluids which involve blood, sweat, tears, saliva, semen, and etc. Okay, so basically it is the study of the reaction between an antigen and antibody. All right. So now, so let's look at parts of the blood, part of our blood. Okay, based on this picture, when you see this picture, you can see that okay, there's a in our blood there are red blood cells, there are some leukocytes here, which is our white blood cells. Okay, so you can see that there are macrophages here as well, which is a part of our defense system, and there is a yellowish fluid, which is a part of our apa ni? It is our plasma basically, right? So there are platelets here, there are macrophages, these are macrophages which are part of our white blood cells. Okay, so red blood cells, or we can call it erythrocytes. Okay, so red blood cells, it carry oxygen, it contains an antigen, and it is the most abundant cell in our body. Okay, so red blood cells carry oxygen and it contains antigen. This is important for you to be able to do blood typing afterwards. Okay, white blood cells, or we can call it leukocytes, which is a part of our immune system. Okay, so white blood cells is a part of our immune system, where sometimes if you heard about T cells, B cells, or even macrophages, so all of that is a part of our white blood cells. Okay, now we have looked at also plasma. Plasma is the yellowish liquid which contains antibodies, proteins, enzyme, clotting factors, vitamin K for our clotting factors, and electrolytes. So all of that are in this yellowish liquid. Okay, and we have platelets as well, which is this yellowish substance here, the platelets, which is for our coagulation for clotting purposes. Okay, all right, so that is a part of our blood. Okay, so how about serum? Serum is actually when we talk about serum is actually the one that is that is corresponds to your antigen and antibody. Okay, we'll look at it afterwards. All right. Okay, so blood typing, a little bit of history of blood typing. Okay, this is not that very important, but it's good for you to know. So blood transfusion attempts often ended in failure due to clotting, because in our blood we have platelets which coagulates clotting processes. This is a part of our body process now. So whenever that, for example, you have an open wound, so our blood, these platelets will usually respond to this by doing clotting, okay, by having a clotting process to make sure that we don't lose a lot of blood. Okay, So by 1901, by Carl Landsteiner, so he created ABO blood typing based on blood factors attached to red blood cells. So this is where we looked at into the antigen of the red blood cells. It allows for a safe matching of blood donor to the recipient. So now when we do blood transfusion, we need to know about the blood type of the recipient as well as the donor. Okay, so knowing this blood type helps narrow down the suspect list. Okay, so for example, when you have found there is a blood at the suspect, which is not the suspect, not the victim's blood. So you have a suspect blood on the victim. So now you can narrow it down to just what is the blood type. Okay, what is the blood type of the victim and what is the blood type of the suspect. So it helps in narrowing it down. Okay. So blood typing. So blood types we have A blood types which corresponds to A antigen. A blood types would have A antigen. B blood types would have a B antigen. Okay. For AB blood types, it will have an A antigen and B antigen. O blood types would not have any antigen. Okay, so you have AB, and O blood type. Okay, so what is a D antigen? D antigen is we call it RH factor. Okay, if the antigen is present, we call it RH plus, or it is absent, RH negative. All right. So basically, D antigen is not that it is important, but it is not that important in terms of our blood typing. So you need to remember AB, and O blood type. And what are the antigens corresponds to those blood type. Okay. So antigens. So as I mentioned earlier, so antigens is a substance, usually a protein, okay, that stimulates the body to produce antibodies to fight an intrusion. So there are usually more than 15 known blood antigens lah. But in our case for blood typing, so you see that blood type A would have an A antigen, blood type B would have a B antigen, blood type AB, which is have A and B antigen, blood type O would not have any antigen. So blood type AB would be called as universal recipient, while blood type O would call as universal donor. Okay. So the fundamentals principle of blood typing is that for every antigen there exists a specific antibody. Okay, so blood type A would have an antigen A and would correspond to antibody B. Blood type B would have an antigen B and corresponds to antibody A. Blood type AB would have both antigens A and B but no antibody. Okay, blood type O would have no antigen but it will have an antibody towards A and B. Okay, all right. So let's look at this. These are the figurative explanation lah. So you have A corresponds to B antigen, B antibody. AB would have no antibodies. So basically, this is we call it universal universal recipient lah. So basically, A and B would can receive blood from A, from B, from AB itself, and from O. But for O type blood O, it will have an AB punya antibodies. Therefore, it only can receive from O. Okay, that's why we call it donor, because O blood can be given to A, B, AB. A only can give to A, B can give to B, AB cannot give to anyone, but only to AB. O can give to everyone. Okay, so that's why in terms of call this type O is a universal donor. Okay, so how does we do blood typing? Okay, we use anti-serum. Okay, anti-serum is to test for agglutination, which is in this case when you talk about agglutination, you straight away would think about clumping. Okay, so when you have anti-A, when we have an anti-A, which is anti-serum A, so it will make blood type A clump. Okay, Because anti-A would have B antibodies. Okay, now we look at anti-serum B. It would have A antibodies. It will cause B, blood type B to clump. Alright? How about O? Type O will not clump with either anti-A or anti-B because it doesn't have any antigen. But type AB will clump with both anti-serum A and anti-serum B. Okay, so blood type AB would have both antigen A and antigen B. So this is how are you going to do the the, the blood typing. So usually, uh, you will imagine that you will have uh, four apa ni four spots lah. So basically you would imagine that you have four blood spots. So you put anti-A, you put anti-B, and you put both anti-A and anti-B. Okay, so that's how you would imagine. Okay, so if your blood when you corresponds to when you put anti-A, it agglutinates, it clumps, so it is a blood type A. Kalau you put anti-B and it agglutinates, then it is type B. Tapi kalau you put type A and type B, anti-A and anti-B and both agglutinates, so it means that it is AB. But when you put anti-A and anti-B and both does not agglutinates, then it is type O. Can you imagine? Right? Okay, moving on. So, how agglutination works? Okay, agglutination. So basically, this is an antibody lah. Okay, this is a part of antibody. But if you want to look this into detail, this is about immunology, right? So antibodies usually we have two binding sites. Okay, this is the light chain. Both of this is the light chain of part of the antibody. This is the heavy chain. Alright, so antibody usually kita ada dua tangan macam ini, where this is the binding site to your antigen. So usually this one side will bind to one, for example in this case one red blood cell. This side will bind to another red blood cell. So it will cause some sort of like a network. So this network will cause a agglutination. Alright. So that's how agglutination works. So in this case, when you look at this is the red blood cell, which is call it erythrocytes. We put anti-serum, it doesn't matter A or B. If it agglutinates, then it will correspond to AB ataupun AB. Kalau O, there is no agglutination. Okay? Alright. Okay. So when you so this is what I said lah. Okay? You imagine you put anti-serum in one spot, anti-A in one spot, anti-B in one spot. Okay? And then you put your blood. You see that during for anti-A, you see agglutination. Anti-B, no agglutination. Straight away, antigen present is A and blood type is A. No agglutination, straight away B and B. Agglutination, both antigens present, therefore blood type AB. No agglutination, is that there are no antigens present, A nor B present, then it is type O. Okay? Alright. So, why do we need to do this? Why do we need to have this blood test? So, forensic scientists must be able to answer this question. First of all, is it blood? When you look at a red liquid at a crime scene, you need to know, is it blood? First question, whether it is blood or not. Okay? The second one, from what species did the blood originate? Is it a human blood or an animal blood? Third one, if the blood is human, how closely can it be associated to a particular individual? So you need to be able to answer these things. Okay. So now we are going to look at on how are you going to test whether it is a blood or not. Presumptive test. Presumptive blood test, we can call it PBT. So there are three presumptive blood tests, which is in this case we can call it phenolphthalein ataupun Kastle-Meyer test, luminol test, and leukemia test. leukemia green test. All right. So when we look at Kastle-Meyer test, so basically, when you look at the picture, when you use Kastle-Meyer test, when it changes to pink, then it is straight away a blood. But you cannot uh, differentiate whether it is a human blood or animal blood, but it is blood. So you have answered the first question, whether is it blood or not blood. Okay. So usually we'll have this Kastle-Meyer solution lah, right? Okay. Leukemia green test pun sama. When you put it, when you test it, it will change to green, then it is blood. So you have answered the first question, whether it is blood or not. Okay. Luminal test. Luminal test is something that you will test in dark, okay, because it corresponds to the fluorescence. Okay, luminal plus blood plus dark will have light. Okay, it will illuminate in the dark. Okay, the sensitivity is extremely sensitive as compared to Kastle-Meyer and leukemia green. So if luminal test is the most sensitive one. Okay, so, but the limitations is that it must be viewed in the dark. The fluorescence only lasts for a while. So usually when you have sprayed onto the luminal solution lah. So you have sprayed it, straight away need to make sure that it is quite dark for you to be able to look at it. Right? Okay. That is luminal test, Kastle-Meyer test, and leukemia green. Alright. So that is all usually on site lah. Okay. So if you want to test on site, usually we have Kastle-Meyer, leukemia green, and luminal. But if you bring it back to the lab, so usually kalau kat lab tu kita guna you can use all of these things as well, but there is another test we call it Takayama test. So Takayama test ni is something it is about hemochromogen crystals. Okay, so hemochromogen crystals form by treating a small amount of blood or a stained fragment. So when you apply the Takayama solution onto the blood, you can see a hemochromogen crystals form. So these crystals are observable under a microscope, which it look like a salmon color crystals, right? So this test doesn't require heating or any, but it can usually be used for older samples, right? So last one in terms of confirmatory test for blood, we also can have a ABA card, Hemotrace ABA card. Okay, this is a confirmatory test, shows that it is a human or a primate blood, since the other tests can be non-specific. So this is the test specifically for human hemoglobin. Okay, but usually usually we use Kastle-Meyer, leukemia green, and luminal lah. Okay, so basically you would have 5 confirmatory five presumptive blood, sorry, not 5 presumptive blood tests, 3 presumptive blood tests. Confirmatory blood tests you have 2. Okay, the Takayama test and the ABA card Hemotrace. Okay, 3 presumptive blood tests, Kastle-Meyer, leukemia green, and luminal test. And confirmatory tests you have 2 Takayama tests and ABA card Hemotrace. So all of this, when you have presumptive test, you can see that okay, this is blood. When you bring it to the lab, you can have Takayama test, so a ABA card Hemotrace as well. Okay. So now we have answered the first question. Now we need to answer whether the blood is human or animal blood. So we can use precipitin test. Okay, precipitin test ni macam mana? Precipitin test ni is a test for you to be able to differentiate between a human or an animal. So based on this picture, if it is a human blood, then you would have a zone of we would have a visible precipitate. Okay, so when we talk about precipitin test, straight away after for precipitin test, if it is a human blood, then you would see a precipitate. Okay, so with that in mind, so how does it goes? Okay, precipitin test, it used to determine if the blood is the human origin. So if it is a human origin, then you have a precipitate. Okay, animals, you need to know that animals will produce antibodies when injected with a human blood. So when we inject a human blood into an animal, this animal will create antibodies, which we call it as human serum. So this human serum can be isolated. Okay, so you imagine that, okay, let's say I put this picture lah. Okay, so you take a human blood from a human, you inject it into a rabbit. So this rabbit would synthesize a human serum. So you will get this human serum from the rabbit and you will do a precipitin test. So how does it goes? So you place a human blood into this test tube, you put the human serum. When you put the human serum that that the rabbit have synthesized, it will create a precipitate. So human blood gives a precipitin band with synthesized rabbit serum. So if you place if you place animal blood into this test tube and you put the human serum inside it, then it would not have any precipitate because only human blood will give the precipitate. Okay, so now you've got in terms of how are you going to determine whether it is a human blood or an animal blood by precipitin test. But it involves the animal to produce the human serum. Okay? Alright. Okay. So can we narrow it down to a particular individual based on this blood type? Okay. So enzymes, which other substance in blood used to usually from the blood. Okay, so usually enzymes in the blood, we can use it to try to individualize the person lah. But usually DNA is the one that you are going to use now. Okay, in terms of how are we going to track down what is the what is the identity of the individual. So usually DNA lah for now. Okay, so so that is for blood. So now, how about semen, sperm, and seminal fluid? Okay, we have semen, we have sperm, and seminal fluid. Semen, sperm, and seminal fluid. You need to be able to differentiate between all of these three. All right. So fluid, white, yellow, of a complex composition, produced by male sex organ is called a semen. All right. Typically, the ejaculation from a male sex organ would consist of 1.5 to 5 ml of semen, which is usually about 40 to 250 million spermatozoa, which is the sperm. Okay? Oops. All right. So this feature of cases of rape, sodomy, attempted sexual murder, usually would presented this kind of evidence. So you need to be able to know on how are you going to detect in terms of how are you going to detect semen, how are you going to detect sperm, and how are you going to detect seminal fluids, right? Okay, so inside the semen, you would have the cellular portion, which is your sperm, and the fluid portion, which is the seminal fluid or prostatic fluid. All right, so usually inside the cellular portion, which have the sperm and epithelial cell, but for fluid portion, you will have a seminal fluid, which is in your seminal fluid. You will see that in your seminal fluid, it will consist of fructose, choline, lecithin, absorbic acid, prostaglandins. All right. In your prostatic fluids, also we have an acid phosphatase, spermine, cholesterol, phospholipid. Right. So all of this you can detect it. All right. You can detect it. So how are you going to detect for seminal fluid? So this is the presumptive test basically when you arrive at the scene, for example, in this case, you arrive at the rape scene. So you want to detect this fluid is seminal fluid or not. So usually we will use a phosphatase acid Britanamine fast blue test. All right. So in this test, they contain a reagent which containing substrate and chromogen is dropped onto a swab on paper. So usually it will, if it is a positive reaction, then it will change to a purple color. So it will form crystals. All right. So but this crystal will give out a purple color. Fresh stains will produce a strong color change reaction. Old stain maybe you have much more weaker reaction lah, because the concentration is not that much anymore. So other materials such as urine, vaginal secretion, and perspiration may produce a weak reaction. So that is phosphatase acid or Britanamine fast blue test, because you are going to test for the fluid portion of the seminal fluid. Alright. Another type of presumptive test for seminal fluid, which is a choline fluorescence test. So in this reagent, fluorescence iodine reagent, aqueous solution of iodide and iodine in water. So the principle is that if there is choline present in the seminal or semen stains when treated with iodine reagent, it will result in the formation of rhombic-shaped crystals of choline periodide. Okay. So false negative due to low sensitivity. So basically, this choline fluorescence, it's the sensitivity is quite low. So this low sensitivity will cause a false negative, right? Okay. Confirmatory test for seminal fluid. So straight away you are going to look at under the microscope to look at for the spermatozoa. Seminal stains is transferred onto a wet cotton swab. So this usually have a cotton swab and swab it onto a microscopic slide. You view under the microscope. So usually you would see that there are spermatozoa. So the head is going to be stained with reddish, mid pieces blue, and the tails are going to look like a yellowish cream. Okay, so this confirms the presence of spermatozoa. In terms of this, when you want to stain the spermatozoa, it is usually by using what we can call it a Christmas tree stain, right? In terms of the staining technique. So these stains would cause this kind of color, okay? All right. Okay. So for seminal fluid confirmatory test, so you would also need, you would also can use prostate specific antigen, which is the most popular one. It targeted the specific antigen. Okay, so what are the procedures? So the swab or cutting agitated in water. So you would drop the solution into the test well. Okay, you put into this test well. When you, I think this is the usual kit. Okay, even though it's a COVID kit pun sama lah kita letak dekat sini. So the liquid will move to the slide, and then we would see the results. Okay, so what are the advantages? The accepted marker, which in this case detecting semen and in criminal cases. So possible in cases where there are no spermatozoa, and you see under the microscope, sometimes it's much more diluted and you will not see any spermatozoa. So, but in this case, when you use this prostate specific antigen, so you can detect it. Okay. All right. So I think that's all for this part 1. So the part 2 we will continue in the Wednesday's lecture. All right. Thank you.